Good morning to everyone and welcome to the second day of the Regions4PerMed conference on socio-economic aspects. So while yesterday we had a technical conference on the topics that we picked for our 5th key strategic areas, today what we usually do when we have this kind of conference, when we used to have this kind of conferences before the COVID, we had in situ visits. So we, with the consortium, we used to go and visit facilities and meet researchers at their laboratories. It couldn't be possible to do this here. I mean, also because there is still, I mean, the tail of the COVID is still hitting us. So we decided to meet in a bigger room called the researchers, scientists from the Tuscany region that are most active in developing innovations and bringing excellence on the territory. And today we are listening not only Tuscany, we also have from Trieste, which is one of our partners. So today we will listen to few examples of the Tuscany and innovation ecosystems, and also from Area Science Park, with which we are collaborating since 2019, I think. So the first speaker is Dr. Cristina Tinti. She's our bioincubation manager in Toscana Life Sciences, and she will be presenting what she has been doing in the last years and the results that we have achieved thanks to her.
Thank you, Gianni, for the introduction. Like a TLS representative, it's really a pleasure, to host this event and also to have the chance to present actually the TLS Foundation. That's what I'm going to do basically today. So let's start. Fondazione Toscana Life Sciences is a non-profit research foundation. Was established in 2004. With a special mission to sustain innovation and applied research in projects in applied research in life science sector with a special focus on health. We have many stakeholders. They are mainly public. We have all the university and the Superior School of Siena, of Tuscany. And then we have the institution, we have the Tuscany region that for us is absolutely one of the prior partner because of the financing part, but also for the tight collaboration in place. And we have also the local institution like the municipality, the province and the Chamber of Commerce of Siena. And then we have two private partners that are the stakeholders that are actually the bank, the Siena Bank, the Monte dei Paschi Bank, and its foundation. Basically, the ultimate goal of the foundation is to improve wealth on the territory of Tuscany by supporting the birth and the growth of entrepreneurial ventures in life sciences, and consequently improve the number of high-skilled jobs. As I was mentioning, the foundation was founded in 2004, but operations started in 2006 with the setup of a bioincubator. Just to reach the first goal, there was the development of a life science cluster in Siena. Basically, what happens, we started with the revamping of a warehouse that we were renting from, at the time, over here there is the campus, a very important campus for vaccines. At that time, there was Chiron. And we, as mentioned, we had a warehouse that we actually started to renovate, and we put labs inside this warehouse and started to implement technological platforms in order to support the companies that were coming in, that were pretty soon moving in. In the meantime, we have also starting to set up services, scientific services that could support companies also in their development process. And on the other side, we have been putting in place all the services functional for the business part, starting from the IP protection, technology transfer, all the way up to identification of the business idea and identification of potential financial partners in order to start the business. What you see here is what we are now. TLS is managing two buildings for a total of 10,000 square meters. We have several labs where companies are located, platforms, offices. And these spaces right now are hosting more than 50 companies and research groups. Some of them are actually residents, some other are affiliated, and we have more than 500 people that are working within these institution companies. And I have to say that these people have a very high education, more than 40% as a PhD, and they are many of them focus on R&D activities, as you can see from the number of publications that have been produced since the starting of our activities in 2007, when the first companies moved in. But what is really interesting to see is also the amount of money that these companies have been able to attract by grants and also private financiers, and the income that has been generated during their activities. Here is the slide of our bioincubator. From the very beginning, we started to have companies that were basically spin-offs coming from the university, the academic context. We have the Siena-Gent, there was Setlan, Siena-Gen, Ricerche, Toscana Biomarkers, coming from all the University of Tuscany. And by the time we have been actually evolving in I would say, a more mature environment, we started to host also spin-off from larger companies, but also labs that were already in the companies, but the companies actually wanted to locate within our system in order to have interaction with the other partners of the ecosystem. So you can also see how the development has been, crowded companies have been moving in, research groups. And then you can see perhaps in 2021 we have Lonza. This is just to give you an idea of the internationalization of our companies. Lonza came here and started to invest in 2019 in one of the companies that established here. And last year there was actually they bought the entire company. And they decided not to take the technologies and move out, but actually to stay in Siena and develop their ideas because of the environment, the very lively environment they could find. Those are the areas of expertise of our companies. We have many companies working in diagnostics, vaccines. Of course, as I was saying, this is since the last century in 1900, the site has been very Sclavo, has been a center for cutting-edge research in development of vaccine, but also in production. Then we have companies that move in, working in agri-food environment, also companies that provide services like NGS, next generation sequencing. Then we have also companies in engineering. And then very important is the group of subjects that work in the oncology. We host the Center of Immuno-Oncology of the University Hospital of Siena. They do a very strong translational research and they apply personalized immunotherapy. We also have an important group working in diabetes. Professor Dotta, that will talk later, we'll talk about their experience. And then, of course, food supplements, and we have also companies working medical device, digital health, and medical hemp. This is just a very quick picture to show you how the ecosystem has been improved. This is the every year we perform a survey with our tenants just in order to understand how they have been growing. And what you see here are the aggregation of the data. And as you can see, very important for us is people. At the very beginning, 2007, when the first companies moved in, we had like 7 people, 10 people. Now we have more, as I was showing you before, we have more than 500 people, young scientists, many young people that are working in these companies. And again, projects, scientific publication, patent application, granted patents. As you can see, there is a trend that is increasing. And very important is also their capacity to generate revenues. As you can see, companies started to generate revenues in 2013, and in less than 10 years, from 3.8, we arrived cumulatively, of course, to more than 38 million. So this is a very nice achievement for us. Besides the incubation activities, that has been the first one that has been developed by TLS, we have been working a lot in those years with the Tuscany region to enlarge the activities also at regional level. So from 2009, we started with the Tuscany region to project implement the Hoover office, that is the office for the exploitation of scientific results that were performed, that are actually performed in the academic context and the university hospital research granted by the university, by the Tuscany region. In the meantime, these activities has been performed with the General Directorate of Health and Cohesion. I don't remember what the number is. Okay, so Health Department of the General Directorate of Health of Tuscany. With the other part, the part related to the business, we have been actually working to implement what is now the the life sciences cluster of the Tuscan region. And within these clusters, we actually gather all the stakeholders that work within the life science sector. So that is research groups, companies, venture capitalists, of course the health system that is very important for all the activities that is run and performed in Tuscany. And we are really trying to enlarge our activities also at national level by working also with within the Alisei cluster, that is the national cluster of Italy. But also we are having many activities at higher level, at regional, at European level, within the bioregion, the CBAR cluster. Another very important activity that TLS has been performing in these years is training. TLS, together with Tuscany region, major companies and all the university, is one of the founder of ETS Vita. This is a foundation highly committed to the training of young people that can work in the life science like technician, very specialized technician. And this is something that is very useful because what we are really doing is capturing the needs from companies and try to train these people, these young people for performing their activities within the companies. And last but not least, as I was saying at the very beginning, TLS is actually a no-profit research foundation, and what I've been implementing recently is some research units that are inside TLS. Claudia Sala, that is here, that is the coordinator of the Mad Lab, will talk a little bit more about it, but what I want to give you is just a broad picture of the three major areas in which we are operating. One is infectious disease, antibiotic resistant epidemics. What I want to highlight, this unit was established in 2018, and in few years was able really to attract an impressive amount of money to perform its science. We also have a unit that is working on immunology. Again, this is a very important aspect that we are following in Siena, also because of precision medicine, the idea to support the clinicians to identify and stratify patients for a better cure. And then, very important, is also the data science lab that has been established very recently, that is supporting all the science that is performed in our foundation. Just to summarize the TLS achievement that we have that we did so far, we have to say that we have been establishing a very lively incubator with many scientists working in it. We have been able to improve our capacity in providing incubation services. we have been reinforcing the technological facility to support the science of our incubators. And as mentioned before, we have been establishing also our own research activities. And we have increased our reputation at national and international level. I want just to finish my presentation, just to give a few hints on TLS and precision medicine, what we have been performing in precision medicine. Basically, one of the most important projects is financed by Tuscany region, and is the establishment of the center, the Regional Center for Precision Medicine, together with the University of Siena and the University Hospital of Siena. Francesco Dotta, that is the coordinator for the university, I believe will talk also about this center. What I just want to highlight is some of the activities of the center, that is the setup of joint public-private technology platforms that are open access. Everybody can access to this platform, use the technologies. We have been also using the money that have been, the financing from the region also to attract research groups, the Center of Immunology Oncology that I was mentioning before. And, but we also have been using this money to support the training of young scientists, PhD, especially in bioinformatics and data science. And another very important aspect is sustained to the research in rare disease. Finally, what's on the scientific side, what we have been actually trying to put in place to support the research in precision medicine, platforms that consent the omics. We have a very strong proteomic unit, and now is increasing also in the metabolomic. Together with the University of Siena, we also are implementing genomics and transcriptomic platforms. And those are very important for the identification of new biomarkers. And then these biomarkers have to be validated. In order to validate biomarkers, we have been establishing this new platform on the generation and production of molecular antibodies that would function as bait, as in order, to absolutely identify these biomarkers in vivo model, trying to validate their function. So this is basically what we are doing for precision medicine. And I just conclude my speech just talking about the future goals and the development. We want to strengthen the Toscan Life Science Innovation ecosystem, basically by potentiating the incubation services and attracting new life science companies. We want to support our companies that have been very successful in, in their scale up and growth, also outside the bioincubators. Very important for us is also the attraction of public but also private funding in order to implement, increase our ecosystem, life science ecosystem. Of course, we want to expand the technological offer in order to support our research groups and companies. Strengthen the territorial value chain with the implementation of a next generation implant for production of vaccines and molecular antibodies. I don't know if you yesterday heard our president during the introduction to the meeting. He was mentioning that Siena, because of his great expertise and tradition in vaccine, has been selected by the government for being one of major stakeholder of the hub against pandemics. So one of the major work that we have to do is actually to put in place, to renew and put in place this platform for the production and be ready and for future emergencies. And another thing very important for us is strengthen, of course, our intramural R&D activities, especially supporting projects in the precision medicine. Thank you.
I don't know if there is any. Yes. Sorry. Okay.
Hello, so I'm Eva from Saxony, and I have been involved in Regions4PerMed as a partner, and my topic was personalizing health industry, and I would just be interested to find out about the financing environment for companies here. So how would you describe the access to venture capital and to growth capital, and also what exit strategies do the investors have here?
Okay, no, very clear, no, absolutely. I have to say that actually when we started with TLS Foundation, we were in a way very lucky because the bank that is one of the stakeholders, TLS stakeholders, created a special fund, a seed fund that was quite interesting for the support of and grow the first startups. By the time actually this fund was not available anymore. And I have to say that we really, especially during the 2010, 2014, I would say there was really a huge bank crisis, so money was not really available. And what happens basically is that the TLS has been, I know it's not so nice to say, but in a way, I have to say that it was a winning solution. Basically started to attract only companies and groups that already had a very good capacity to attract money. So this was in the period, I would say the middle period in which we really started to crowd the incubator. And I have to say that these companies has all been very, very good, performed very well. Probably their idea was so good that they were already able to attract personally private venture capitalists. They also put own money. And by the time we have been strengthening our action through also the life science cluster, and we have been making many relations with what are the VC, major VC in Italy, and not only in Italy, and also with all the, I would say, public venture capitalists, let me say, like we have Cassa Depositi e Prestiti, we have some ventures that are actually financed by the government. And what we have been doing basically is make this sort of network, and every time a company requires money, another very important aspect actually is what also Gianni is helping us to set up, that is the identification of important grants at European level and financing by European Innovation Council. So this is another aspect that we are really trying, to put in place. And what happens when a company requires money for their growth, we basically put them in contact with all these venture capitalists and the financing institution that we know. And basically, this is a process that we accompany, but we are not directly involved. For the exit strategies, this is a big question because so far we didn't have exit strategies because the problem was that we never were able to implement what for us was the final program, that was the post-incubation, meaning building an area and for the growth of the companies. And because of for the stakeholder, the crisis of our stakeholders, the bank crisis, we couldn't really, perform this project. So what we have been doing basically is just to maintain these companies within the bio incubators, and they grew a lot, and this is now a real problem. So this is what we actually are planning to do, is finally that our finance are a little bit better to build, to build new spaces and to let this company move out and have their development over there.
This is the exit from the incubator, but I was also thinking about how do venture capitalists exit the companies? I mean, what opportunities are there for growth companies? So, for instance, I think it's an issue all over Europe that there's no real stock market for small companies in the life sciences field. So I wonder, what is the approach that you can observe here? Is it been an issue?
I have to say, for what is our experience, the few venture capitalists that have been investing, they are still within this company. So I cannot tell you exactly what could be. I don't know if you, Gianni, have some clue on this topic, but so far these investors are still within the companies. But, I have to say that we have only 2 companies that have venture capitalists that have been financing. All the companies, as I was saying before, have been financed by private capitals business angels, and also many money coming from the European Innovation Bank and European Innovation Council grants. So I have to say that for the number of groups and companies that we have, the weight of investments is really low. It's a lot of private investments. So I'm sorry I don't have the slide with me, but it's really nice to see how during time the income generated by the companies has become the major source for financing the further development of the companies. This is what has been happening in Siena in our model.
So you have observed self-sustained growth, I would think, yeah?
The idea was also, exactly, not by TLS, it's companies, they generate so much income and they are able, at a certain level to invest in innovation and in new products. But again, there is this very strong network also with the government, what are the public financing, the grants. And so there is also the Tuscany region has been very, very innovative in the tools that has put in place to finance companies. So far I have to say again, on 50 tenants that we have, only a couple have VC that are investing within their companies. So
how many companies are there that are developing medical entities? Because I saw this, and there was in the beginning there were quite a few, and then it went down, because I think this is what needs most of the money.
Things, no, what is happening, this is a diversification. There are many diagnostic tools, so that's the reason why there are services. I have to say that there are not only drugs that are the final products of companies that are working. I have to say that actually that the most successful companies in our network are services companies. They provide services. And I have to say, in a way, unfortunately, but the COVID for those companies was a real opportunity. Many of them had the chance to really grow.
Thanks, Eva. Is there any other questions? Okay, thanks a lot, Cristina. So I'll move on with the new presentation from Dr. Claudia Sala. As Cristina mentioned, she joined TLS in 2018, and she's the head of the Monoclonal Antibody Discovery Laboratory, very active during the pandemic. And I'm very happy to say that a few weeks ago, I think, she received the award of Cavaliere Ufficiale al Merito della Repubblica Italiana, which is one of the biggest awards that the President of the Italian Republic gives to, in this particular case, for the work that she and her lab did for the fight against the COVID-19. Claudia, the floor is yours.
Thank you. Good morning, everyone. And I join Cristina in thanking the organizers for inviting me to share our experience with you all. So, as Gianni said, I'm responsible for what is the MED lab. So MED sounds as MED, but also as Monoclonal Antibody Discovery Laboratory. It's a lab which was born actually at the beginning of 2019, when Dr. Rappuoli received the first ERC grant, because he wanted to tackle one of the major problems of today, which is antimicrobial resistance. And then from there we moved on, and we became as big as with 25 people we are today. So that's, now it's an old photo, because in the meantime some people left, some students graduated, and others got their PhD, and so they took their own careers, and others joined. You see Dr. Rappuoli sitting in the middle, so he's our PI. He's the person who has inspired and continues to inspire our activities. So the MED lab includes people with very diverse expertise. I myself, I'm a molecular microbiologist by training. I spent quite a long time abroad, Pasteur Institute in Paris and then EPFL in Lausanne, before moving back to Italy, where I was looking for something that could complement my CV. And I was looking indeed for Professor Rappuoli, who is a world authority in vaccinology. And then some other colleagues are immunologists, some are mathematicians, informaticians, some others are chemists, We are also very different in terms of age. Some of us are seniors with some experience abroad. Others are young students at the beginning of their career. So it's an integrated group, which includes a very diverse expertise and a different way to approach science. So our mission basically can be summarized into a couple of sentences. First, we would like to tackle one of the major problems of global health today, which is antimicrobial resistance. Here we are talking about bacteria, so those bugs which cause pneumonia, urinary tract infections, but also hematological problems sometimes. So we have several bugs in our pipeline, and we are trying to address this major problem, which consists basically in the lack of efficacy, unfortunately, of most of the antibiotics that are available so far. And the other point is targeting SARS-CoV-2, so the virus which stepped into our lives a couple of years ago, and it's still around. And so the way we decided to follow in order to address the two problems is to try and find monoclonal antibodies for therapy. So we are not looking for antiviral drugs, we are not looking for antibiotics, but we are looking for monoclonals, so very specific and targeted molecules. And in this way, I would say this fits perfectly into that framework of precision medicine you have heard before. In addition to using monoclonals for therapy, we also would like to use them as baits for fishing the cognate antigen, so that this can support a rational vaccine design. In this slide, you see a summary of our projects. In the middle, you see the 3 bacterial species we are working on, this Neisseria gonorrhea, Klebsiella pneumonia, sadly famous, as I will explain in a minute, Shigella. Here on the side, SARS-CoV-2, and there on your right-hand side, mRNA. mRNA because it's the cutting-edge approach nowadays. So it's the way to go, and we are also exploring this avenue in order to make our monoclonals available, not just as recombinant proteins, but also as mRNA counterparts. So for today's purposes, I will focus on antimicrobial resistance. Antimicrobial resistance was flagged by the WHO some years ago as one of the biggest public health challenges of our time. Indeed, it has been estimated that every year we have more than 700,000 deaths, and these are supposed to increase, and by 2050 they would even bypass the number of deaths which are caused by cancer today. So if you think of these numbers, this is something to be really frightened of, and it's time to take action and develop something which could be a game-changing strategy into this. So the WHO clustered the bugs that you see listed here into 3 major groups. The inner one, which is the critical one, includes 2 of them, Klebsiella pneumonia, as you can see in the red box, and Escherichia coli. So 2 of the bacteria which cause most of the nosocomial infections so far, and most of the infections which are increasingly difficult to treat. Then in the high priority list, we have Neisseria gonorrhea, the cause of gonorrhea in humans, and finally in the medium circle, the upper one, you can see Shigella. So Shigella, which causes several diarrheal diseases, especially in children under 5 years of age. So what can we do in such a situation? Well, one could say, well, discover new antibiotics. That's easy, you know. Explore the chemical space, try and find new molecules which can target these bacteria. But the other point is one can develop vaccines. But again, this involves a number of important steps, both scientifically and on the market side. And here in this slide, I explain where the problem is with antibiotics. So basically, I think we can say very fairly that the golden age of antibiotics is nowadays over. So there has been the golden age of antibiotics in the 40s, 50s, 60s, when most of the molecules have been discovered and have been developed as new medicines to treat bacterial infections. But then as we move towards the end of the millennium and then the beginning of the year 2000, no more chemicals were discovered, and indeed the chemical space is now reduced and there is a little list of new pharmacophores that can be exploited for drug discovery. On the other hand, you see the green line with the number of vaccines, which is steadily increasing over time. And nowadays, we can say that we are probably in the golden age of vaccinology. And monoclonal antibodies actually have something to do with vaccines, as I will explain in a minute. Another mini digression on what vaccination is. So all of us, we have been through vaccines against COVID-19, you know? And so we should know how important vaccines are when you are in the middle of a pandemic. Indeed, here you see the comparison between antibiotics and vaccines. On the top part of the graph, you see that basically all of the antibiotics that were discovered, and you see the yellow star, sooner or later they became useless because resistant species came up and they took over. And so even the very last ones, which were discovered, like linezolid, daptomycin, and others, we already have resistant bacteria around, and these resistances are spreading. So we need to carry on and continue this game of humans against bacteria, against humans, against bacteria, and so on. And this is going to be quite an indefinite story. On the second half of the slide, you see that vaccines were discovered, again, where the yellow star is, but none of them generated resistance, actually. There are a couple of them with the blue cross. That means that in those cases it is difficult to tackle all of the serotypes which cause the infection caused by these bacteria, but actually there is no resistance to vaccines. So this is just to compare the two approaches. But there is some time, as I said, and I think I stressed it enough by now, when antibiotics become useless, and so one has to explore new avenues, and among these, we certainly have monoclonals, as I anticipated, but also consider antivirulence compounds. For instance, in the area of tuberculosis research, this is being done. And others are the host-directed therapies, which are a complement to antibacterial therapy. So for today, I will talk about monoclonals against infectious diseases, and here present a table which comes from the review cited there, which is 5 years old, but still very good. So there are some advantages in developing monoclonals. What are these? First, monoclonals are extremely specific medications, so meaning that you can target very specifically the bacterium you want to address. And so you can spare the microbiota, for instance. On the other hand, if you use antibiotics, you kill everything, you know, unless you are so lucky as to find a very specific antibiotic. Sometimes monoclonals are the only option for immunocompromised patients. For instance, when people cannot be vaccinated for any medical reason, you can administer monoclonals. We can benefit nowadays for the enormous technological progress which consists in high-throughput cloning and expression of monoclonals, and also a lot of efforts have been put in engineering the monoclonals in order to improve their penetration, effector function, even conjugation to drugs. So today we can make antibody-drug conjugates which exploit the specificity of the monoclonal and the potency of a drug, for instance. So you can deliver locally, thanks to the specificity of the monoclonal, a drug which otherwise would go everywhere. And this has, as you can see, enormous benefits because you can reduce probably the amount of drug to be administered. Anyway, next to the advantages, you see some drawbacks, some flaws that still need to be addressed. So for example, what is the accurate animal model for testing monoclonals which are supposed to go in humans then? The problem of antigenic heterogeneity of pathogens. So bacteria are very smart, smarter than us, and so they like masking themselves behind capsules, behind robust cell walls. And so if we have to cross those barriers, we have to recognize those barriers which are very frequently diverse. This is a major challenge that we have to face. So as I say, the capsular layer may mask important antigens. And last but not least, what's the precise timing for administration? Should we give the monoclonal in a prophylactic way or as a therapy? So these are all open questions that are being answered, I would say, nowadays. A few examples of antibacterial monoclonals. As you can see, again, taken from the same review I mentioned before, there are indeed some which have been approved by health authorities, such as the FDA in the United States, but most of them target the toxins. So it's a very specific targeting against a very specific virulence factor, meaning that, for instance, for Clostridium botulinum, we know that its own toxin is responsible for the disease it causes, and so efforts have been directed to finding monoclonals against that. So up to the year 2018, 2020, there have never been efforts directed at finding monoclonals, I would say universal monoclonals, able to target most of the antigens of a specific bacterium. You may wonder what a monoclonal does to bacteria. Actually, the story is a bit different compared to what an antibiotic does, because while antibiotics usually target essential functions of the bacterium, and so by inhibiting these essential functions, you kill the bacterium. In the case of monoclonals, the story is a bit different. So in the first panel here, you see the so-called neutralization, which is a physical blocking of either the virus or the bacterium in these cases. The monoclonals bind, and so they prevent the pathogenic action. In the second panel, you see agglutination. So monoclonals can promote this aggregation of bacteria in this case, and this in turn recruits specialized cells, such as the macrophages, which are component of the immune system. They could be compared to the waste truck moving on into our body, and so they go around, they control what's around, and they pick up whatever bad they find. And then in the third panel, you see the binding to specific antigens. So we could imagine that those yellow circles are the toxins we mentioned before, and monoclonals can bind and precipitate them, and so they make them not active anymore. And finally, one of the most important functions carried out by antibodies is the activation of the complement system. So meaning that antibodies can talk to other components of the immune system, and so recruit these components to the site of infection and act in a way that bacteria are then killed. So the complement is a series of proteins which act one after the other, in a subsequent activation, and at the end, what they do is making a hole on the bacterial cell, which then explodes and is dead. So I will focus on one example among the many that we are considering in our lab, and it's about antimicrobial resistant Klebsiella pneumonia. So that's an outbreak which was initially recognized in Pisa, which is not very far from here, so we are in Tuscany. And there are a couple of citations from Italian newspapers. It's in Italian, but it means that there is a superbug in Tuscany, which caused at least 17 deaths. So the issue took place initially where you see those colored circles there in the northwest of Tuscany, so Pisa and the surrounding areas. So it means that in those areas, clinicians started in 2017, 2018 to observe some increasing cases of Klebsiella pneumonia infections. And the major point here is that these bugs were absolutely refractory to any treatment. There was no antibiotic that could kill them. And so the clinicians had no option to treat these patients. And basically they hoped that the patients were so strong, so their immune system would be so strong to resist and overcome the infection. Microbiologically speaking, as I am a microbiologist, I must introduce the bug here. So Klebsiella pneumonia is a gram-negative encapsulated bacterium. It's an opportunistic pathogen, meaning that sometimes it infects people who are already weak for other reasons. You see here in this scheme that there is a sort of coat all around the bacterium, and this coat, which is called capsule, can be more or less thick. In some incredibly highly pathogenic strains, this capsule is very thick. These are the hypercapsulated strains. In some other cases, the capsule is a bit less thick. However, it's still present and can protect the bacterium from the action of the immune system. Klebsiella belongs to the group of the so-called escape pathogens that you can see listed there on the side. In addition to Klebsiella, there are some other bugs: Enterococcus, Staph, Acinetobacter, Pseudomonas, and Enterobacter. So together they constitute, let's say, the 1, 2, 3, 4, 5, 6 most challenging bacteria which cause opportunistic and nosocomial infections. What's special about the outbreak in Pisa is that these bugs carry a gene which confers resistance to most of the so-called beta-lactam antibiotics. So antibiotics which kill the bacterium by inhibiting the cell wall synthesis. And this gene encodes for the New Delhi metallobeta-lactamase, NDM. New Delhi, so the name tells you the origin when it was first discovered. And so we have now this bacteria which can transfer this resistance mechanism from one to the other. And so this increases and expands the problem exponentially, as you can imagine. So the first thing we did when we got to know this, we decided to get the bugs here. And so we had 30 isolates from the hospital in Pisa. We sequenced them by high-throughput sequencing, and the analysis told us that basically it was a clonal population. So 29 out of 30 belong to a single group, that group 1 in red there, which is known as ST147 or sequence type 147. So it means that these bacteria look basically all the same. They are very similar. This is why I say it is a clonal population which expanded in that area. Only one of them belonged to group 2. You see ST11, sequence type 11. So it is, and it was, a little bit different, meaning that the face of these two types of bacteria is different. So if we want to target both, we need to find monoclonals which can recognize both types of bacteria. Okay, that's what I mean when I say the face of the bacterium. So as we all have a different face, also bacteria have a different face, and so they show different membrane, different capsule to the immune system. And that's what Klebsiella pneumonia shows. So Klebsiella pneumonia shows a capsule, which I said can be more or less thick. The capsule is composed of sugars. Basically, it's a very, very sweet bacterium, I would say, but very dangerous. In addition to the capsule, we have the so-called LPS, the lipopolysaccharide, which is a component of the outer membrane of bacteria. And again, it's a complex molecules that you can see outlined there. It is anchored in the membrane thanks to the lipid A, which takes it into the membrane. And then we have, again, repeating units made of sugars. So it's a super sweet bug, but it's not very tasty if you try it, you know. So we need to find something to tackle the infection caused by this bug. So what's the take-home message? Obviously, I don't want you to remember all of this, but the take-home message is the complexity of the bacterial surface. And so we need to find cross-reactive monoclonals in order to be able to address all of the instances that come up. And so in our lab, we applied this strategy, which I'm describing now, for the isolation of monoclonal antibodies, in this case against Klebsiella, but also against other bugs. So we start from patients, in this case recruited in Pisa, who had bacteremia, so had the bacterium in their blood. And then we isolate monoclonals, or more specifically, we isolate their cells which produce monoclonals. So we isolate the B cells, which are specialized for producing monoclonals. Then we cultivate these cells in vitro. We get them to produce the monoclonals, which are then tested for binding and for activity in several functional assays. So we go from the in vitro to the ex vivo to the in vivo in order to be able to capture all the possible activities that monoclonals can exert against the pathogen. So we could have the CDC, the complement-dependent killing that I mentioned at the beginning. We could have the AIA there, AIA, which is the inhibition of adhesion to an epithelium, for instance. We could have inhibition of biofilm formation. We need to validate for sure the activity of the monoclonals in vivo, in the mouse model, or whatever else is more properly useful. So all of this takes us to proposing monoclonals either as therapy or for antigen discovery. One thing that we should not forget in this incubator, which is TLS, is that we are contributing to developing new tools, and especially tools based on microscopy. Nowadays, watching films as they happen is key in science, okay? So we have a powerful and high throughput, high content microscope, which allows us actually to develop new tools which allow us to see what's happening to the bacterium. And this is important to understand the mode of action, and also to understand, if you want, how to combine, for instance, different monoclonals into a cocktail therapy. So to summarize what we have done so far, we recruited 9 patients, more than 21, also almost 22,000 memory B cells have been isolated. We selected more than 500 monoclonals able to recognize Klebsiella pneumonia, and out of these, a bit more than 200 were expressed. Then these 200 underwent the screening that I described before, and now we have a short list of 25, 28 monoclonals. Here you have only 15. And for each of these, we tested the ability to bind the different Klebsiella strains, because, as I said, each of them has a different face, and so we need to test if they can bind each. So we did see that some of them cross-react versus different Klebsiella strains, which is good, because we are looking for a kind of cross-reactive monoclonal or a universal monoclonal. And so this led us to hypothesize that probably the cross-reacting ones can recognize conserved structures on the surface of the bacteria, and now the trick is, which is this conserved structure? Here is one example of what I meant when I said confocal microscopy. You see examples of 3 monoclonals binding to 4 different strains. The first one on top is what we call eye binder. You see how it decorates very nicely the surface of the bacteria. The second one binds a little bit less nicely. In some cases, you see dots, and not all of the bacteria are decorated by the antibody. The third case is even worse. Here we have an extreme specificity, in my opinion, for targets which are not abundant, but are indeed present on the bacterial surface. Then after the characterization of the binding, we need to move on with the activity, and here you have the full list, I believe, of the 30 or so monoclonals that we selected, with their activity against the different strains on top. We measured the IC50, which is, the unit here is nanograms per ml. So you can see that in vitro we are well below the nanomolar range, which is extremely potent for bacteria, and actually compares these monoclonals to the potency of the best antiviral monoclonals which have been described so far in the case of SARS-CoV-2. So that's all we have so far. Where are we going now? In vivo validation, that's the first thing, and for sure we will try and benefit from what Christina described before me, so the facility present in TLS and the capability to carry out this kind of experiments. And last but not least, target identification, both for rational vaccine design and for designing cocktails of monoclonals, which could be useful for targeting more than one serotype of each bacterial species. I believe that was my slide. Yes, my last slide. So thank you very much. I'm happy to take questions.
Thanks a lot, Claudia, for this wonderful presentation. I don't know if there is any questions from the audience. I have, okay, Go ahead, then I have a question.
Okay, so since I have a personal background as a biochemist, so that's why I also have a question to the drug that you're planning itself, I mean the monoclonal antibody. This is something that is not probably going to be used in a very broad population, but mostly in hospitals where you have severe infections, I suspect, because you have to administer it into the blood, into the systematic system. You cannot, I mean, antibiotics that we know, they are usually, many are taken orally, and that's why they've been prescribed so easily, and that's why we now have resistance. But I mean, how will you do a study to get this to the market? Because you have very low patient populations. Is it then served as an experimental drug? And in fact, in the end, when after you did all the preclinical and toxicity studies and phase 1.
So okay, so on the administration route, first I must say that the intravenous infection is not the only option nowadays. So in our case, I didn't mention it, but we did develop an antibody against COVID-19, which got to phase 2-3, and then it was unfortunately interrupted because Omicron stepped in, and it made all our monoclonals ineffective. But anyhow, these monoclonals could be administered intramuscularly. So if this is the case, it means that even a doctor at home can do it. There is no need to go to the hospital and undergo a long treatment with IV infusion or this type of things. So in the case of antibacterial monoclonals, that's also what we would like to do. So we would like to reach the possibility to go IM, or even delivery by aerosol. That would be the dream. For example, if you have the upper airway infection, it would be great if we could design a delivery system that could take the monoclonal directly where it is necessary, so by aerosol. So that's about the administration route, which is one story. And then the little cohort, this is true, but unfortunately it's not so little because the outbreak is still ongoing. And we are writing today a research proposal in collaboration with colleagues in Germany where a similar outbreak is being experienced. So unfortunately, I must say, we expect this outbreak to grow bigger and bigger. And so there will be many patients where the antibody can be tested.
I also have a question. Actually, two questions. One is, when do you expect this to be ready? I mean, granted that the preclinical studies will be successful, to be ready for the clinical phase, and if you have an estimation of the investment needed to also go into clinic.
The honest answer is no. To both. Meaning that, well, we have seen, again, I must cite COVID because it's our most benchmark, thank you for the word, benchmark experience. So the story can be very fast. It can go very fast as 4 months, if we have funds and if we have all the regulatory in place. But it can be delayed if all of this is missing. So increasing the awareness, also in the civil society, increasing the awareness and let me use this word, lobbying a little bit with those who make the decisions for funding. It's extremely important if we want to give relevance to this. But the major point here is increasing the awareness of this silent pandemic, as we call it, which is antimicrobial resistance. Because nowadays we are focused again on COVID, and we don't consider that antibiotic-resistant bacteria are indeed the challenge, not the next challenge, but the challenge which we are already facing. And so that's the major problem, and both at the regulatory and financing level.
Okay, for the funding, I think this is a problem for Christina. So the other question is, what is within the TLS ecosystem that accelerates, that helps your research group to speed up the research? I mean, if there is any, also starting from the experience of COVID, what are the key success factor that you think TLS can offer as an ecosystem?
In my opinion, it's freedom.
Okay. Oh, yeah.
Because, TLS is not a company, and it's not an academia either, but it has the benefits of both. So since my return from Lausanne and from France, I thought that this place could have the added value of putting together what the academia can give you, which is the freedom of conducting the research you want, and to have the relationships you want with collaborators and partners abroad. So I think encouraging this networking and relationship is one of the keys, and something that we can do here very well with the support of many colleagues. Some of them are here in the audience. And the other point is also the advantage of being a private staff, so we can speed up some decisions, and that's also key. I mean, you don't have to wait for the delays sometimes of the public things. So these are the 2 points that I see most clearly in TLS.
Okay, thanks a lot.
Yeah,
I don't know if there's any other questions from the audience. Okay. Oh, there's. We shouldn't answer his questions.
No, I don't want to take too much time, but it was interesting for me to know more about going back to antimicrobial resistance. What about phages?
Phages, so
Yeah.
phage therapy, yes.
Yeah.
Phage therapy is a super exciting thing that comes and goes. There are some golden months for phage therapy and some months when it's not considered anymore. Yeah, I think it's a super interesting. I have never worked on it myself, but I remember also when I was in Lausanne, and my boss did consider it as a potential anti-tuberculosis therapy. However, I think it has more or less the same challenges as monoclonal antibody discovery, because phages can be very specific to a specific strain of bacterium, and so you would need to identify a phage which is as broadly active as possible. So probably the challenges are more or less the same. Yeah. There are examples indeed in the literature where this is brought up, but as I said, it's not really my area of research, so don't feel that I can comment more. Yeah.
I just have one more question. Another field of activity that you've described, just very briefly, that's the mRNA maps.
Yeah.
So what exactly are those aiming at, and what you're thinking about in this context? Is it also, yeah?
So we have a project in the lab whose aim is try and deliver monoclonal antibodies as mRNA molecules. So meaning that we would exploit then the human body as the factory which makes the antibody by using the instructions which are brought there by the mRNA. So it's the same principle as the mRNA vaccines that most of us got against COVID-19, exactly. So what did we get against COVID? We got vaccines as mRNA molecules, and then we made the spike protein ourselves by using the instructions given by the mRNA. That's what we would like to do with the monoclonal antibodies. So we inject the mRNA into the people, and then these people would make the antibody, and so you would benefit from an amplification system, because from each mRNA molecule you probably translate more than one antibody molecule. That's what we would like to do.
You would, for instance, make muscular cells take over the function similar to B cells.
I'm sorry, say it again.
So you would have muscular cells, for instance, if you take over a function like B cells for a while at least. Mm-hmm.
Yeah.
Okay.
Wonderful. Thanks a lot again, Claudia. Sorry? Scusami, non l'avevo vista, ma la riprendiamo al coffee break. Okay, so our next speaker, I think Cristina introduced him, and this project is Professor Francesco Dotta from the University of Siena, who is the responsible of the Regional Center for Personalized Medicine, and he will be describing us and presenting us his work. Professor Dotta, can you hear us?
Yes, clear.
Yeah.
Good morning,
Screen is
everybody.
yours.
Yes, apologies for not being able to join you there. Let me share my slides. Let's see. It says that I don't have any slide, but however, I have my PowerPoint presentation. Can you see the slides?
No, we can't.
I
Wait.
have no idea how to manage.
I'll just.
Maybe if you look on your screen on the bottom left, you should be able to see like 2 different squares. Did you share your slides?
Yeah, I do, but it doesn't. It says that there is no slide.
Okay, hold on a second.
Let me see if I can. Let's see if I can. Let's see if probably I can upload.
I was just speaking Italian.
Presentation.
Francesco, mi dicono che non stai condividendo, però.
In realtà sì, sto condividendo It says that I can share the slide.
No,
It simply says that there is no slide.
Solo un secondo. Ah, forse we are sharing.
Professore, dovrebbe andare in basso, c'è un più, condividi, slides.
Lo vedo se riesco a vederlo da qua. Let's see if you can.
Tuo computer.
Yeah. Hopefully you are there now. Can you see the slide now? Yes.
Yes, we can.
Yes, now we can.
Great. Sorry for this. So again, good morning, everybody, and apologies again for not being able to join you there. So today I will going to present some of the activities of the Regional Center for Personalized Medicine, and in particular, what we are doing in this center in the field of diabetes as an example of the disease that can greatly benefit from a precision medicine approach. Of course, when we talk about precision medicine, I think a major boost was received in early 2015 when President Obama, in the State of the Union address in January of 2015, raised the issue about precision medicine, and in particular, he mentioned cancer and diabetes as a change of paradigm in terms of medical approach to certain diseases. And as a diabetologist today, I will show you an example of how precision medicine can be of support in stratifying and treating better our patients. Nowadays, we know that a single individual can be characterized not only in terms of his genomic fingerprint, but also from several other points of view, starting from the microbiome, environment, proteome, transcriptome, and so on. So overall, we have to start to think about in terms of integrated omics for health and disease. So we are now able to generate a huge amount of data from the several aspects that characterize a single individual. The problem is that now we need to integrate all these databases that are coming from different fields in a sort of what we can define as a sort of integration engine. So different competencies are necessary to achieve this approach. And so we have to start to consider, and in an academic environment now is something that has been accepted somehow, that both scientists, clinical researchers, physicians, and patients on one side should be part of a measure system that should be, let's say, used and optimized to design clinical trials, specific treatments, to design guidelines and specific diagnostic tests to overcome specific diseases. With this in mind, thanks to a generous contribution of the Toscana region, here in Siena, as Cristina already mentioned before, it was set up the Regional Center for Personalized Medicine through interactions of 3 different institutions. On one side, the University of Siena, on the other side, the Azienda Ospedaliera Universitaria Senese, namely our university hospital, and finally, the Toscana Life Sciences Foundation.
Francesco, can you hear me?
Yeah.
Non vediamo le slide. We can see the slide moving.
Oh, you are
still on the first page.
Sorry. Slide. What about this? Can you see the slide now?
No, we can't.
I surrender. I really, I don't know how to overcome this.
Solo un secondo, Francesco. Okay.
Let's see now.
Okay. Now we can see. We can see them moving. Okay, thanks.
So basically the concept, I'm sorry for this. Basically the concept was that now we have to think in terms of different actors, such as scientists, clinical researchers, physicians, and patients on one side, to act all together and in order to develop new treatments, to design appropriate clinical trials, to develop guidelines and specific diagnostic tests. So, thanks to the generous contribution of the Toscana region, a regional center for personalized medicine was established here in Siena. And in this center, there are 3 main actors. On one side, the academia, as the University of Siena. On the other side, our university hospital. And last but not least, the Toscana Life Sciences Foundation. And the strategy behind the Regional Center of Precision Medicine is to share platforms, expertises, and competencies in order to boost collaborations between different actors. So on one side, we have a series of open access technical platforms, and I'll show you some of them, and Cristina already mentioned some. The second, to set up biobank that are in terms of tissue, cell, and not only tissue and cell biobank that are characterized by shared standard operating procedures for sample collection. To share bioinformatics and big data management, and in order to promote translational research and biomarker discovery, all finalized to, for example, design targeted trials or virtual clinical trials. So this is a list of some of the open access technical platforms that are available in the Regional Center for Precision Medicine. Here we have the nanostring encounter analysis system, the slide scanner nanozoomer, ultra-sensitive multiplex biomarker, the NovaSeq, as well as other next generation sequencing apparatuses. We have a triple, quadruple mass spectrometer, Muldi-TOF spectrometer, an Orbitrap mass spectrometer, the FT mass spectrometer for exactly biopharma. We have a deep array, laser capture microdissection, the Biofax Canto cytofluorimeter, Confocal Microscopy, the Opera Phoenix high content screening system, as well as additional equipment such as quantitative PCR, the droplet digital PCR, and so on. So with this in mind, we know that biomarker discovery is one of the main topic of and the main aim of what should be a regional center for precision medicine. So we apply this approach to a specific disease, which is diabetes. Diabetes, as you all know, is a chronic disease, is a heterogeneous disease, and when we have a precision medicine approach to this type of disease, we have to take into account different aspects, starting from the phenotype of the patient in terms of demography and the presence, for example, of comorbidities. On the other side, the genotype that not only can influence the susceptibility of a given patient to diabetes, but also, for example, can influence the pharmacokinetics or the pharmacodynamics of a given drug that we use for that given patient. And then we have characteristics of the disease in terms of disease duration, severity of the disease, the presence of fasting or postprandial hyperglycemia, the characteristics of the insulin secretion, and finally, the characteristics of the drugs in terms of pharmacodynamics, pharmacokinetics, and pharmacodynamics. So among the candidate biomarkers that we are studying, I will present very briefly what we are achieving in terms of the analysis of microRNAs. MicroRNAs, as you know, are small non-coding RNAs that are involved in the gene regulation. In general, it's a negative gene regulation through transcriptional inhibition or mRNA degradation. MicroRNA are now a class of molecules that are deeply involved in several cellular processes, including cell differentiation, proliferation, and apoptosis. And a specific and interesting characteristic of microRNA is that Micrornas can be deregulated at level of cellular as well as tissue level in several diseases. An additional and probably more interesting characteristics is that microRNA not only are present at cellular tissue levels, but can also be detected in biological fluids, including plasma, urine, saliva, and so on. And in these biological fluids, they can circulate either free or coupled to HDL, for example, in the plasma, or also part of the secretory microvesicles or exosomes. With this in mind, we can say that microRNAs can represent potential excellent biomarkers since they are very stable in clinical samples, such as plasma, for example. To measure circulating microRNAs, I mean, the technique is minimally invasive, and they can be taken as candidate biomarker for disease diagnosis, prognosis, or treatment response. And so we decided to apply this technique in a specific disease, thanks to our participation to a major European consortium that study type 1 diabetes. Type 1 diabetes is that form of diabetes, is an autoimmune disease characterized by the T-cell mediated destruction of pancreatic beta cells, so the cells that produce insulin. And this is a disease that once used to be defined as juvenile diabetes or insulin-dependent diabetes. Juvenile diabetes is no longer used since type 1 diabetes can occur at any age, although it represents by far the most frequent form of diabetes in the pediatric age. So we decided to apply this approach, the approach of studying circulating microRNAs in type 1 diabetic, in a cohort of new onset type 1 diabetic patients, to understand whether we could stratify patients based on this biomarker profile and whether microRNAs could be used to predict the beta cell functional decline. In other terms, whether through the measurement of specific microRNAs, we can predict whether the beta cell failure, the failure to produce insulin, can be predicted or not. So the cohort we used is a cohort of type 1 diabetic patients at onset. More than 100 patients were analyzed and were followed up for up to 2 years. And at diagnosis, we obtained samples, plasma samples, that were subdivided into 2 aliquots because we analyzed microRNA profile through 2 different platforms. One is HDG mRNA at SEC, and the other one, the small RNA sequencing platform. So these are the patients. There were overall 116 patients, 100 in the pediatric age, under the age of 18, and 16 aged older than 18, all within 4 up to 6 weeks from disease diagnosis. And these are the 2 platforms that we analyzed. The first one is HDG, which is a targeted sequencing approach using specific miRNA protection probes. In this system, we can analyze up to 2,000 circulating microRNA using such a small volume of plasma. Just 15 microliters of plasma are necessary to have a complete microRNA profile. However, with the limit of this system is that you can analyze only the 2,000 microRNA that are available in the system. In addition, we used a small RNA-seq analysis platform with the NovaSeq 6000. In this approach, basically, is a totally unbiased type of analysis in which you can analyze all non-coding RNAs. And with this system, the volume of plasma which is necessary is 200 microliters of plasma. So using both platforms, we analyze this microRNA through a method that is highly standardized and that was published a few months ago in the STAR protocols, in the STAR protocol journal. And this is the follow-up of our patients. As you can see from this slide, we can divide these patients at the beginning were characterized by a certain amount of insulin secretion. Clearly, with the follow-up, there are two different groups of patients. The first group that progressively lose insulin secretion over time. So the beta cell destruction probably is ongoing and is more and more aggressive. And the other group which maintains a certain level of C-peptide progression, C-peptide secretion. In other words, insulin secretion. That means that there is a great deal of heterogeneity. And in case you have to design a clinical trial, you have to keep in mind that probably you have 2 different populations in front of you. And if we analyze through the 2 different platforms, those 2 groups, we can clearly discriminate with the signature of microRNAs the progressors from non-progressors in terms of beta cell damage. So microRNA signature at diagnosis can be associated to a future loss of beta cell function or not. But what is also very interesting is that if we characterize the fingerprint of these 2 groups of these patients through microRNAs, regardless of the beta cell function profile, you can clearly see that there are 2 different clusters of patients that are clearly evident using both platforms that we call cluster A and cluster B. These 2 clusters are not different in terms of gender distribution, age, hemoglobin A1C levels, so these are the glycemic control, fasting blood glucose level, fasting insulin secretion level, body mass index, or insulin dose at diagnosis. However, for example, cluster A is characterized by higher level of insulin autoantibodies, while other diabetes-associated autoantibodies such as anti-GAD, anti-IA2, and anti-Zin transporter 8 titers were the same. And insulin autoantibodies are usually associated to a more aggressive type of beta cell destruction. And interestingly, the two clusters differed in terms of insulin requirement 3 and 6 months after diagnosis. In other terms, through a specific analysis of microRNA at diagnosis, we could predict somehow the insulin requirement 3 and 6 months after diagnosis, while the insulin requirement after 1 year was different. Again, reflecting probably these two groups of patients at different pathogenic mechanisms. And looking more specifically to some microRNAs, it is interesting that 10 microRNA belonging to cluster A are all together in a specific gene locus, which is located in chromosome 14. And this chromosome 14 is indeed a type 1 diabetes susceptibility locus, raising the possibility that some action is taking place at the given chromosomal locus in which different microRNAs are located. And the other microRNAs that are upregulated instead in cluster B include some inflammation-related microRNAs that you can see listed here. While in 16 upregulated microRNAs upregulated in cluster A include insulin secretion related microRNAs. So on one side, we have microRNAs that are upregulated and are involved in regulation of the immune response and inflammation. And on the other side, we have microRNAs that are involved in beta cell function. And finally, we try to correlate the fingerprints of microRNA to other omics and specifically immunomics in collaboration with King's College, which analyzed the very same patients in terms of immune profile, analyzing more than 150 different immune cell subsets. And what we found is that, for example, the two clusters differed in terms of Th17 positive cells, CD8 made positive cells, as well as CD4 T effector memory cells. Again, underlining that these two different clusters identified through microRNA fingerprinting differ in terms of immune response. So in summary, through this approach, we basically could establish that circulating microRNAs at baseline can help in stratifying type 1 diabetic patients into different endotypes that we call cluster A and cluster B, which are characterized by different titers of insulin autoantibodies at baseline and showing different insulin requirement profiles at follow-up during type 1 diabetes progression. Then we identified a set of microRNAs that weighted more in the distinction of these two endotypes, and specifically those that are located in chromosome 14. And finally, that microRNA highly expressed in cluster B are mostly involved in innate immune and peripheral inflammation, while those hyper-expressed in cluster A showed enrichment in insulin granule fusion pathway and insulin integration signaling. Again, confirming that there is a great deal of heterogeneity of type 1 diabetic patients that can be uncovered using a microRNA approach. So let me finish thanking the whole team that collaborated with this work, with this effort. As I mentioned, The Type 1 Diabetes Consortium is called Innodia, is a huge European consortium funded through the Innovative Medicine Initiative approach. And these patients are followed up and usually can enter specific clinical trials based on their characteristics. Because the problem that we have in the scientific community of type 1 diabetes is that so far, most of the clinical trials failed because the concept was that type 1 diabetic patients were identical among them. This is not true. There is a great deal of heterogeneity. So if we want to be successful in preventing beta cell damage, we have to keep in mind exactly that there is this heterogeneity, and we have to stratify our patient as much as possible to be successful in protecting their beta cells from the immune-mediated damage. Thank you for your attention.
Thank you very much, Professor Dotta. I don't know if there is any questions from the audience. I have a question, but it's more like from the ecosystem perspective. So you mentioned that TLS is involved in this project in the Regional Center for Personalized Medicine. Which are the ties, which are the links between your institution and TLS and Tuscany region for the implementation of the Regional Center for Personalized Medicine?
Yeah, thank you for this question, because what I showed you is exactly an example of how the shared facilities can be successful in achieving a specific aim. The CREMEP, the Regional Center for Precision Medicine, indeed allowed us, for example, to participate successfully to a series of competitive grants, because altogether we were able to put different expertises that were a key for success. Personally, I can say that thanks to Toscana Life Sciences Foundation, I was able to do research in the last 15 years, I would say, 10 to 15 years, because the concept of sharing facilities and projects is a winning strategy if you want to to do good research at international level. And another thing is the multidisciplinarity that is present in TLS and that university and academia appreciate very much. So the strategy of thinking as a team rather than a single person is something that is a positive characteristics of our environment, and the Toscany region recognized this. It is not by chance that most of the funds were, settled here in Siena, because there were three different entities, such as University, TLS, and the University Hospital, that all together were sitting around the table and sharing strategies and aims.
Perfect. Thanks a lot, Professor. I think we can. No, sorry. There's a question from the audience. Go ahead.
I just have a question that was very interesting, because it shows that you have this very wonderful infrastructure to use to identify different and categorize different patient subpopulations. Now, what does it take to do this for a larger patient population? Because not all hospitals are equipped like you are, and not all diabetes centers. This is the one question. And how long would it take that patients can benefit from findings such as these? What do you think?
Oh, thank you very much for both questions. The first one is that now this consortium is, is based on in 28 different centers in several European countries, and there are satellite centers. We have now almost 500 patients that have been enrolled, plus approximately 5,000 over their first-degree relatives as a population of patients potentially at risk to develop the disease. All these patients are characterized in depth for several omics. Here in Siena, we characterize for microRNAs, but for example, the same patients are characterized for immunomics in London and Cambridge, for autoantibodies in Helsinki, and so on. So all these biological samples are shared among the community. In Italy, we have a network with several centers in Rome, Milan, Pisa, in addition to ourselves, and Chieti, that are already part of this network, and similar networks are based in several countries in Europe. As far as the, I would say, clinical transferability of these results, there are already 4 clinical trials that have been that are ongoing through clinical trials developing different immunomodulators that in which patients are enrolled based on the very specific characteristics. These are new onset type 1 diabetic patients. Hopefully in the future, we would like to enroll also high-risk patients before they develop the disease. But the status today, yesterday actually, we have a meeting in Brussels on to make the point on this consortium, is that 4 clinical trials are already ongoing in which patients have been stratified before entering the trial.
Okay. Thanks a lot. Any other questions? Perfect. So I think thanks again to Professor Dotta for your time and your presentation. I think it's a time now for Professor Paolo Milazzo from the University of Siena. University of Pisa. Will be presenting the Biomedical and Health Informatics Lab.
So, good morning, everybody. And thanks to the organizer for inviting me to give this talk. My name is Paolo Milazzo. I'm a computer scientist from the Department of Computer Science of the University of Pisa. And I'm here to present you, to say a few words about the Biomedical and Health Informatics Lab of our department, so of the Department of Computer Science of the University of Pisa. In Pisa, we have a long tradition in computer science. Okay, in the 50s, we had the first Italian supercomputer that has been constructed there. Then, in the 70s, we had the first Italian internet connection in Pisa. And then, as a university, we had the first department in computer science, the first degree in computer science at the national level. And this tradition now brought us to have, in our department, a lot of research groups that cover almost all of the disciplines that are that compose computer science. Okay, because computer science is quite a complex set of sub-disciplines that often are not distinguished by the non-specialist, but that require very different expertise. And many of our groups are involved in life science projects. So they apply their methods in the context of life sciences. And we realized that in many cases, in order to be successful in applying our methods in life sciences, we have to collaborate with our colleagues. And so we decided to create a network of research groups inside our department in order to be able to create synergies between the different expertises, and also to be able to become a single entry point for the external stakeholders. So in this way, we can present to the outside world as a single entity but with many expertise inside. And when we have to participate in a project, we can participate together, okay, rather than as individual groups. This is the list of research groups of the department that belong to the laboratory. Okay, we have the research group on algorithms for genomic sequence analysis, the biosystem modeling group, that is the group I'm coordinating, the group on computational health, computation intelligence and machine learning, but also cyber physical system and the health. So they are experts on development of Internet of Things devices and so on. As well as data sciences for sports analytics. We have a specific group and also a spin-off that works on sport analytics, which is mostly related actually with the aspect of health. Home care services optimization. Also ICT methods for precision agriculture, which has many overlaps with methods that are used in the context of health. And also the very big group of knowledge discovery and data mining, which is actually, as I said, a very big group that work on big data analytics in general. And we also have great computing facilities. We are quite proud of our green data center, which is, I can say, something unique, at least at the national level, for university at least. It is a Class A AGID data center. It has a 250 dedicated building, also quite far from the university department, okay? And we have a lot of servers and computational facilities and an efficient cooling system that allow us to call it a green data center. And this is a very important resource, even because this data center is freely available to all of the faculty of the University of Pisa for their research need. If we need computing facility, we only have to ask, and we can get them quite easily. And altogether, this shows that in our lab we have the competencies and we have computing resources, and this makes us, I think, an interesting competence center in the context of computational methods for life sciences. Just to give you an idea of how different can be the computer science disciplines that can be involved in the same project, I'll show you a couple of examples of research projects in which we are involved. The first one is PerMed 2, which is a research project focusing on type 2 diabetes. It is funded by the Tuscany region. We are involved together with the University Hospital of Pisa and the University of Florence. And the idea here is to collect data from patients that could develop type 2 diabetes and try to develop a predictive model, okay, based on those data. It is quite a classical, how to say, precision medicine project. And the idea is to collect data from physical activity using smartwatches or smart bands that many people have. Collect data about dietary habits, conducting surveys among the participants, and also collecting data about the metabolic profile through blood samples. Okay, these are the most relevant data sources that we are considering. And then we have to integrate data and try to make predictive models. Okay, so from the viewpoint of computer science, here we have several disciplines that are involved. For instance, the collection of data from the smartwatches requires experiences with wearable devices, experiences with cloud services that can store data, and so on. The use of the dietary data requires experience on data analysis, clustering, machine learning, for instance. And analysis of the metabolic data requires bioinformatics experience. Okay? And these four aspects, these four disciplines require expertise that in our departments are in 4 different groups. Okay. And now they are collaborating in this project. This is another example. This is actually a project which is currently submitted for funding to the Italian Ministry of Health. It involves most of the university and university hospitals in Tuscany. Actually, University of Pisa, the university hospital here in Siena, and the university hospital in Florence, and also the Toscana Life Sciences Foundation. And here, the idea is to integrate data about different diseases in order to find, how to say, common mechanism. Okay, the fundamental idea is that we would like to try to deal with diseases, with the disease as a collection of biological mechanisms that cause the disease. And the idea is that we could integrate data of different diseases to find a common mechanism. And the identification of common mechanisms could lead to, for instance, to repurposing of drugs. Okay, and also could lead also to better treatment of patient-specific, disease situations. Okay, and again here the computer science disciplines that are involved are quite different because we have to create the infrastructure to collect the data. So again, we need expert in databases and cloud services. We have to analyze data using bioinformatic tools. Okay, so we need bioinformaticians, and of course we need also experts in machine learning, artificial intelligence in order to make predictive models. Again, okay. So again, another example of a project in which we need the integration of different computer science expertise. So the first message of my presentation is this one. Computer science is quite a variegate discipline, even if this is not often perceived from outside. And if you want, experience in all of these disciplines, you need to involve research groups that are focusing on those topics. And this is what we are trying to do with our laboratory. Then, since the topic of this conference is about ethical aspects and socio-economical aspects of precision medicine, I decided to say a few words about what are the ethical aspects of working with patient data. That is something that we have to do in order to perform computational analysis in this project. And as you can imagine, ethical aspects when you work with data are primarily related with privacy issues. Okay, we are speaking about patient data, and so they are subject to privacy regulations, which are quite strong. Okay, and require attention. But this is not the only point that you have to take into account when you work with patient data, and also when you analyze patient data, okay? Because you do not only store patient data in your server. You perform analysis using algorithms on those data. And also this is a topic that raises some ethical issues. Because actually, when you profile a patient, when you stratify patients, or when you try to use some artificial intelligence method to make a prediction and suggest the physician a possible treatment for a patient, the patient has to be aware that you are using this data in this way, but also has the right to get explanations about the prediction and the suggestion that your algorithm provides to the physician. Okay, so this is another delicate aspect, ethical aspect that has to be taken into account when you deal with patient data. Let me say a couple of words about both things. So, as regards privacy and data protection, we know that we have GDPR that now is established since some years. So we know how to manage patient data. We know that we have to do to use pseudonymization and anonymization techniques. We have to pay attention on where we store data. Okay. Often we do not think about what we do with our computer, and we save some files on a shared folder, let's say on a OneDrive folder or a Google Drive folder, without taking into account that when we do this, that file will be copied into a server that maybe is in the United States. So data that we save as a file and as an Excel file in a folder that appears to be in our computer actually are automatically moved outside of the European Union, and so this makes this action not compliant with the GDPR regulation. And also, privacy requires you to take care of who can access the data. So when you deal with patient data, you have to be sure that you store them taking into account cybersecurity issues, okay, that now are quite crucial. In these days, the University of Pisa is under a cyber attack. This happens quite often, actually, okay? And so you have to be sure to take all of the measures that can protect your data from this kind of attacks. But you also have to be able to provide data to those persons that have to work with them. Okay, I have to say, I think the GDPR introduces the data protection officer as the expert person about how data have to be managed, and often the DPO pays a lot of attention on avoiding data to be misused. In the sense that focus on acquiring the approval from the data owner in order to the data to be used and so on. But the right of finding a good treatment is also a right of who provides you the data. And sharing data between experts that work on therapies allows better cures to be found. Okay, so there is this balance to be found about protecting data and allowing researchers to do their job in order to find better cures for the society. And even locally, inside a single entity as a university, who is responsible for the data has to take into account that there are these two forces that have to be counterbalanced. Okay, so sorry for the fact that we have our own data center makes our life easier because we do not have to send data outside our institution. Okay, and I can say that in research project this simplifies a lot of steps that have to be done in the first months of the project. You do not have to make agreements with external data storage services. Data are with you, and you do not have to worry too much about this. As I said, another ethical issue is the right to explanation. Okay, when you use an algorithm to profile a patient and you suggest physician the best treatment for a specific patient, the patient has the right to get an explanation for such a prediction, for such a suggestion. And this is not trivial, in particular when you use artificial intelligence methods to make such a prediction. Let me try to explain what's the problem with an example. What you typically do in this case is to start from a training set of patient data that you collected, for instance, in clinical trials or on project or from hospitals and so on, in which for a specific pathology and a specific treatment, you know that a selected number of patients with certain genetic profiles or dietary habits or so whatever features you have to consider for the pathology responded or not to a specific treatment. Okay, this is your so-called training set, the data set you can start from in order to construct the predictive model. Then you take this data and you feed some artificial intelligence model like an artificial neural network. This is one of the most common machine learning models that used nowadays to perform classification in the context of precision medicine, for instance. And the idea is that, roughly speaking, an artificial neural network is depicted there on the right as that network, as that graph. In each node of the graph, you have a mathematical function that has a number of parameters. You feed one by one the data of the patient from the left. The mathematical function compute results that are passed from one level to the other. In the end, you get a final result, which can be, let's say, 1 or 0, and that represents the success, so the fact that the treatment, the patient respond or not to the treatment, okay? And then in order to perform a training, you feed the network one with the data of the patient one by one. You compute the result. If the result corresponds to the label that you collected, so the information you already have about the success of the treatment, okay, you continue with the next patient. If the network computed the wrong result, you go back and you change the parameters of the functions, okay? This automatically, okay? This is machine learning, so this is the act of learning from the neural network. You change the parameters so that now the result that you compute is correct, and you continue. If you have enough examples, if you have enough patients for which you know whether the treatment was successful or not, in the end, the parameter that you find allow the network to be accurate in making predictions, okay? So if you have a new patient for which you didn't start the treatment, you want to decide whether to start or not, you can provide the data to the network, and it will tell you, okay, I think, based on my experience, that the treatment will be successful or not, okay? But the point is that the functions that are inside the neural network are general purpose functions. They have nothing to do with the disease, okay? And so if the network tells you, no, this treatment will not work, you have to explain to the patient why you are not going to give the treatment to him or to her, okay? And this is not trivial, because you cannot say, I'm not giving to you this drug because the parameter 25 of the 4-node means that computes 0 in the final result, okay? Because it makes no sense to the patient. So what we need now are new machine learning methods or artificial intelligence methods that can be explained, okay? And this is quite a hot research field now. The definition of methods that can tell you why they took a particular decision, proposed you a particular result. And for instance, the information that you can obtain from these methods here is the role that each feature of the patient had in taking the decision. So this method now can tell you, okay, I think that the patient should not be treated with the drug because, for instance, he is too old. Okay, and statistically, this parameter causes the treatment not to be successful for this kind of patient. Okay, and this is a way to tell the patient something that is a sort of explanation. Okay, and in our laboratory, in particular, in the KDD group, we have this group is one of, at the European level, one of the top groups working on explainable AI, and we have several research projects that are undertaken in these days to produce methods that can provide explanation to predictions. So, second message of my presentation is that, okay, we have to take into account privacy when we deal with patient data, but we have also to take into account the right to explanation when we use, in particular, artificial intelligence methods to analyze and make predictions based on data. Third message that I would like to send to you is about the environment in which we work. Okay, we are in Pisa, but we interact with other entities. You have seen from the project that I've shown you before that we work with Siena, Florence, with university hospitals, TLS, okay. And actually, we believe that the regional level is particularly suitable for undertaking this kind of projects, in particular in Italy, okay. And this for several reasons. First of all, in Tuscany, we have a lot of facilities that allow us to create a successful life science ecosystem. Okay, we have universities, we have hospitals, we have higher education institutes, we have TLS, we have industries. Okay, but also from my personal viewpoint as a computer scientist, the fact that we work at the regional level is particularly provides a lot of advantages because the kind of data that we can work on is, I mean, the data are rather uniform, okay? Because patients are all subject to the same regulations, because in Italy, the health system is regulated at the regional level. Patients are subject more or less to the same administrative procedures, okay? Data are collected at the regional level, okay? So, for instance, the Tuscany region provides a service for anonymizing patient data, providing you a unique regional identifier. So if you collect data from about the same patient from different sources, you can cross-reference them quite easily because they have the same identifier. Okay? And also we have agencies that maintain centralized data for all of the Tuscany citizens. All of these make the life of the computer sciences much easier because you do not have to deal with incoherent data or data that have been collected in different ways. Okay. But data are homogeneous, but they are also, in many cases, enough. Okay, because depending on the pathologies you study, very often you have enough patients to carry on studies in a reasonable way, okay? Or at least you have, since we have 3 big universities and a lot of big hospitals, you can exploit collaboration, international collaboration of the people working in the different institutions in order to collect enough data to perform meaningful projects. So we're very happy with the fact that nowadays the region is investing in strengthening the collaboration at the regional level between all of these institutions. And this is my final takeaway. Thank you so much.
Thanks a lot, Paolo, for this wonderful presentation. I have a couple of questions. Which kind of, I mean, when in your data center, which kind of health-related data do you have? I mean, you have the data that you have collected from the project. You are able to take data from other sources like ours. I mean, which kind of health-related data do you have?
We have data from research project, for
okay.
sure, that can, mostly the first project that came to my mind are projects that are done in collaboration with our hospital and also with IRCS Stella Maris. We have a big project with them and all of the data are stored in our data center. Data from us can be accessed without the need of the use of the data center. It's
like
quite
the federated data approach, like you go there.
Yeah. It's quite easy to. Ours provides them. If you have a project and you have good motivations, it is not difficult to ask them for the data. And we have room for more data. Actually.
You also have classified like for pathology, for disease, or
no, they are project related.
Okay.
They are
yeah.
not all integrated together.
And you say that everyone can access this data. Does it mean that anyone from University of Pisa also, or third parties from outside, like other researchers from
No. Anyone from the University of Pisa can access the data center. So I can, if I need a server I can ask for it and can get it. Then data are related with project, and so data are not freely visible to all of the research in Pisa. But the fact that they are inside the university allows us to if needed and if complied with the aim of the project to collaborate easily, to extend the collaboration easily.
Okay, thanks. I don't know if there's any questions from the audience. So I have another curiosity. So you said that you are open to work. I mean, from the health perspective, how do you intend to? I mean, where do you see the area of development? Like, are you going to collaborate more with on health research projects so that you can feed these databases with data and then like make other research on your own? Like, what's the vision?
I mean, our aim as a university is to do research, okay, and to develop new methods, actually. The data centers and the creation of databases is a way to be able to interact with other people in order to face new problems that require new methods. Okay. And so the kind of collaboration we look for, a collaboration that allows us to that give us motivations for doing new things from the research viewpoint and to develop new methods and so on. We are quite open to use the data center as a way to allow this research to be undertaken.
And do you work with companies? Like, there are like artificial intelligence companies all around Italy or Europe that with whom you cooperate to develop these new methods for?
In some cases, yes. Not really in the context of life sciences.
Okay.
We collaborate with industry from the pharma sectors.
Okay.
So that provide us problems that which we develop methods.
Okay. No worry. That's good.
I wanted to say thank you for this very interesting talk, and I also would like to comment that I think it's a very good development that we get explainable artificial intelligence, because I think it's something that is very scary for people if they cannot understand what's happening or what is important for the data analysis. But I have a question. So what you said was really interesting, that in the regional context you have a good environment for doing research. And this is helpful for many indications, but of course, when you go to real personalization or to rare diseases, then maybe the regional focus is even in Italy too small. And so, first of all, are there any initiatives within Italy to interconnect regional data exchange that are, are there any approaches to support that? And the second question is, I know there's European legislation that is supportive of more data exchange in the health space, the European health data space. So have you taken note of that, and what do you think?
Yeah, thank you for both questions, because they're both very, very interesting. About the rare disease, the regional level is maybe the national level is not enough for dealing with rare disease, but I make you a concrete example. Here in Siena there is a group working on alkaptonuria, which is a rare disease. We started to try to collaborate some time ago, but we didn't not work together actually for the moment. But the point is that if you have in your region like in this case, a group which is recognized as an excellence on such a rare disease, you can exploit the fact that from the connection of that group, you can collect the data worldwide to work then with them. So in this case, the regional level is useful because you are close to the group that has the competence on that disease, and that can be the pivot, the point of attraction of data from the also the groups abroad. Okay. Instead about the sharing of data across Europe and those initiatives, yes, an important aspect there from the viewpoint of computer science is the way in which you share data. Okay, now since a few years, the principle of fair data is becoming crucial. The fact that you arrange your data in a way that they are findable, that you can easily, that you add annotations and so on using standards. We have a project on this aspect in particular to work on practice to make our data fair in order to be able to share them more easily. And I have to say that in Pisa we also have we decided to create a center that aggregates all of the big research infrastructure, and so all of the facilities that are used by biologists and also physicists to perform their experiments are the biggest one, at least, are inside the same administrative infrastructure. And the data center and this center that are now collaborating in order to make all of the data that are collected through those instruments stored in the data center in a fair way in order to be easier to be shared. We are working on this because from our viewpoint is the point that requires more work in order to be ready to share as now it is expected to be done.
So, but still, are there Italian initiatives to interconnect data regionally to have certain standards?
I'm not aware of initiatives of this type, but
as they said yesterday, it's easier to collaborate at an international level than within the regions. No, but it is true. Thanks a lot, Paolo.
Thank you to you.
Okay, so our last talk will be given by Dr. Danilo Licastro from Area Science Park. He will be presenting the Laboratory of Genetics, Genomics and Epigenomics, the so-called LAGE. We have invited Dr. Licastro because TLS has an agreement, a collaboration agreement with the Area Science Park, thanks to Francesco Senatore, who coordinated this collaboration. So, Danilo, the stage is yours, maybe.
Thank you, Gianni, and thank you for all. Thank you, the organization, to invite me, to invite us as Area. And indeed, yet I'm working in a genomic and epigenomic lab. I'm the head of the lab. It is actually an open lab. So the meaning of it is that we are aiming to create collaboration. We do not often have our own research goal, but we collaborate for others to using the infrastructure. Just a few slides to introduce what is Area Science Park. Area Science Park is a public entity. We are 100% funded by the Ministry of Research. We do get some funds from other ministry and obviously from competitive grants. We are mainly located in Trieste, Friuli Venezia Giulia region, and we host many other institutes since we own 2 campuses. The one that you saw in the picture below, the one in Basovizza, is where also my lab is located, where you see this big arrow. But as you can see, there's many other things going on, especially there's also the molecular accelerator there, and we are the major shareholders of the molecular accelerator. while in the other campus in Patriciano we host companies and the research institute, just to mention the International Center for the ICGB is the Italian site of the ICGB is hosted there. So it's a pretty nice environment in the middle of, yes, a wood. But this, I mean, it's fun because we have so many different kind of expertise. There's a lot of physics going on. What is probably really missing, and something that I envy in Toscana Life Science, is such a stronger relation with the physician and the clinical in the clinical studies actually. We did get founded in 2019 to extend our some of our places around Italy, and this is the reason why, as Gianni said, in 2019 we started an agreement with Toscana Life Science actually on personalized medicine. It was at the end of 2019. We didn't know what is going to come. But I'd like also to remember that we have collaborator is based on the so-called hub and spoke idea of finding knowledge and activities around, and to share the platform among the different members of the spokes. We are also aiming to international partnership. For this reason, we recently applied to be part of the CERIC-ERIC, so the Central European Research Infrastructure Consortium. We are just waiting to finish our application to be part of it, so we will provide access to the platform through the CERIC-ERIC, and we are also applying to be a node of the Elixir Consortium for the use of a biological platform. When I first received the invitation for this Congress, I went straight back to the director and say, I'm a little bit confused. I'm working in the lab. I'm not really dealing with the ethical, economical, legal. Actually, the legal aspects are the one that I try to avoid. Maybe something for the public trust. So what do you want to be the message that I deliver in the Congress? And the answer was pretty straight. It's what's your perspective? What's the lab perspective? And so I will try to talk about how this ethical, economical, and public trust is actually somehow always present in everything that I do, and also how we decided to do things as they are. As I said, we are a genomic and epigenomic lab. We established the lab in 2004, not as Area Science Park, but as a consortium where Area Science Park had was one of the major shareholders, actually the bigger shareholders. And then in 2018, 2019, we decided, ARIA decided to take on board the lab, so make it directly under the ARIA structure, and we got refurbished. So we saw lots of technology changes in the field of next generation sequencing from 2004 to 2019. Now this is the final setting, so we are set mainly on this commercial provider of sequencer that is called Illumina, as many of you will know. So we are a certified service provider for Illumina. We do roughly hundreds of whole genomes per year, but we do a lot of genotyping, so data production. But this is pretty funny. I do remember that 10 years ago we were talking that those kind of data will disappear thanks to the NGS, and we are still producing for the epigenetic part. Actually, thousands of samples have been processed in a year in our labs. And another funny part about it is that if you strictly look at the machine that I'm showing here, none of them were think for clinical application, but only for research. And even many of the reagents that we use, there's only one exception that is hardly ever used for it, that is the DX series from Illumina. But as I said, how we reached this decision, how we decided to go there, and actually how, as a scientist, I've gone there just on a hinge, and why, since I use it a lot, but as a public entity, that was not enough to decide which kind of technology to use. And one of the main driven of this kind of decision were basically the economical aspect, the economical value. First of all, we knew that the market, the segmentation on the term of end user, put the research center just below the hospital and clinics. So one of the big questions for us was why we need to do so, and not the hospital and not the clinics, because it's it should be the major end users. And the other one was okay, the kind of use was on drug discovery. This is a segmentation of the genomic NGS market actually, and only the diagnostic segment is the second one. But the trend is that, or at least that was the trend in 2019 up to the 2025. The trend was predicted to be really in a steady increase, and this was before the pandemic. Now it's even higher the level that. So economically, it was something that need to be done, and to provide this kind of access to this kind of technology. And the idea on how to choose the technology was to cherry picking something that was at the moment of the choice well established and with a lot of portfolio of activity. So Illumina, that is almost well known in the market since more than 10 years, is definitely one of the choice, and this is why we got the NovaSeq sequencer. But we decided also to bet on something else. And actually, on the first part of the graph, in the technology that are not so well known, but are really potential. And this is the reason why, for example, we decided to go for single molecule technology like the Oxford nanopore. That was the decision in 2019. If I have to think how the economy is going to influence our activity in the lab, this is what is really going on right now. A battle, a furious war about different companies. Just to make you an example, Illumina banned the BGI distribution of the sequencer in US. It managed to do also in Europe, but just a month ago, something like that, the BGI basically had this new awards, so they need to be refunded of the money. And the whole war is raging around the fact that sooner or later, actually sooner than later, the patent that he's holding for the Illumina technology is going to last. They've been the lead in the market for more than 10 years, but it's most likely that due to the expire of this patent, we will see new technology, or at least it's what we believe will happen reaching the market or reaching our market. They are already present in other market, and soon, not so later, that will also reach the way how the genomic in precision medicine will be really acting. So yes, maybe the topic of the Congress was not so far of on the activity of the lab, but I wasn't sure, and I say okay, but they focus on patients, and we built the whole idea of the platform around I dare say older idea of the One Health. So I'm really thankful to be here in Toscana Life Sciences, because thanks to the microbiology field, it's pretty easy to understand the One Health. I mean, it's obviously for maybe for the people of the Mad Lab, so it's pretty easy. But this is how we built the platform to be open to industry and research. So we do accept also requests from research, and we worked on many different fields. So yes, we do a lots of work for the human health. There you see some of the publication that we have been involved, but there's also a lot of clinical activity that is not related to research. We do a lot of exome, we do a lot of whole genome sequencing. We did some non-invasive prenatal testing. We built some predictors on that, but we have done the same more or less also in the field of animals for livestock. That many companies asked for livestock activity based on genomics, and we have done the same also for bacterial identification, and also in some case also for TLS. And then all the platform was hijacked due to the COVID-19. So basically, at the start of the pandemic, the ministry start asking us to focus our attention in all the things that can help in the COVID-19 research or whatever they can be. And so we started doing COVID-19 sequencing that for us, for this kind of platform is pretty easy. I mean, if you compare to a human genome, it's really tiny genome, and we need tons of samples compared to the one that we usually run to make it properly working. But this is how we started working for the local hospital, for the treatments of patients, to identify the strain. We were the first to identify the whole sequence in the region. We have been part of the GSAID group for the surveillance going on and forth. We managed to work also with the TLS group in some cases, and then we open also to other countries that had needs to sequence something, because we were somehow lucky. We do it every 15 days for the region. We can also we see the effort of the other regions. So in Italy, we do sequence. We decide to sequence only few of the positive sample during the survey, but it's a health driven and decided issue. So there's also plenty of space to do more of what we have already done. And for this reason, we helped also other nations like Iraq, Dominican Republic, Ethiopia, Moldova, and also we are now trying to do something for establishing a lab of sequencing, not only for COVID-19, but also for flavivirus in Africa. And it also helps us for using some technology that we had in mind to start using, like the single cells. This is an example of a single cells that we have done on a long COVID patients with the classification of the cells and the presence of the COVID in which kind of cells. So this is hopefully something that is under review. We hope that it's going to be published soon. Again, this is not our work only. It means that there's a bunch of scientists and the physicians that do the works in the real hard work in the field. It wasn't enough, so the ministry said that it wasn't enough, and we need to grant access where we did provide all the library preparation, all the reagent for the sequence. We did the sequence and we did the analysis for on a call, Italian call that is called Fast Track COVID-19, where we basically co-founded all the project in this way through the platform as I said, for research, so mainly university, but also a bunch of hospital. And what is also interesting for us is that we did also for industries, and everybody from Italy were able to apply it if they want during the time frame, and we granted around 20 projects. Some of them are still ongoing but the only question that we ask, the only requirement that we ask were about, it must be about COVID, and the data must go public as soon as possible in an open way. And this is actually one of the main topics that we that we still legally need to face, because the genomic and epigenomic lab is only one side of the platform. The other one is exactly the data center, like the one that we just show from Pisa talk. Is more or less the same idea. A petabyte of space, lots of CPU and GPU to make the user doing their calculation there and access there. But the problem was that we asked and we designed things to be fair by design. I'll go quickly here because it's already been introduced what is a fair, fundable, accessible, interoperable, and reusable. And this is how we decided from our side to handle all the data that are produced in the lab. So this will help a lot to make things reusable if we design from the start, keep the fair approach in mind. Findable and accessible is something that is somehow out of our mind, because it's of our hands, because it's related on the database where we need to put the data on. The COVID sequence is a perfect example. Is pretty findable and accessible for me, maybe not for all of them, but it's also the way that they already have decided to use and to store the data. So we won't enter there in that particular case, and this is something that is not a problem only for us, but also for the other countries in Europe. For example, the G8, where all the COVID sequence and the flu sequence are normally uploaded, is actually not completely 100% accessible. You need to comply with their requirements, and this is not how the fair that we designed was actually working. and is not so findable either, because they're doing not such a great job in keeping the data there. But the idea of find and reuse other data is pivotal for us. I mean, all the data mining and meta-analysis that we do are based on this idea. If we need to compare hundreds of similar experiments, why repeat them if we can mine from public available data? If we need to compare our data with what has been already published, like when we do meta-analysis, like in this is a case of bulk NGS compared to a single cell. I don't want to enter into detail of it, but the point here is that we need this kind of data. We need this kind of structure of data to do this kind of analysis. And actually, is also, we need it to sequence. The new technology helps us to sequence in real time and to analyze in real time. It means that if we have access to the data, we can decide while we are sequencing if what we are sequencing is exactly what we want, or if something that we can avoid and keep sequencing. I don't know how many of you are used to the Oxford Nanopore technology. Is the third generation sequencing that I just described at the beginning, but it's compared to the next generation sequencing, you don't have the data production and after the mapping, you can do it in real time. This means that while you are sequencing through the pore, if you have data, you can check with the data you are producing, and you can, since those are long reads, you can decide, okay, this is reads of my interest, keep sequencing, or no, it's not the sequence of my interesting, pop it out and let another molecule enter the pore for the sequencing. So, but all the, sorry, the slide caused me somehow overlap, but all these requirements, all this fairness to use this data in our hands, often when you use precision medicine data, or we ask for data through the hospital, crash against the GDPR. And usually the reason why they crash so much, and this is an example of few collaborators that highlighted this point, if the purpose limitation of the point of the GDPR principle. Because if you want to strict to the definition, if they were not foreseen in the purpose of the GDPR, you cannot use it. So either you make some big effort to cover as much as you can imagine they will be used, or it's otherwise it can be really the bottleneck of how to use the data in the fair way. So they should be somehow entangled and integrated. We perceive as something different. From one side in the research, the fair approach, and the other side in the clinical case, the GDPR. And so far for keep away from the legal aspect. Just the last few slides, just to make fun. I'm sorry the slides are in Italian. I'll try to translate, but it was about the social aspect. And this is a real call that we got in the lab roughly a month ago or something like that. They started like, good morning. I'm calling for me, I'm calling to book an exome. They were talking about exome sequencing for me and my wife. I was from the other side of the phone and say, good morning, I'm sorry, maybe there's a misunderstood. We can receive a request only from the hospital. My physician told me that it could be a good idea, and you are so good. Thanks, but we cannot receive sample from private citizens. So no, I suggest to go consulting medical genetics from the closest hospital. But we are fine, we're in good health. We don't need an hospital. The sad part about these things is how the communication went completely wrong somewhere, somehow, and what is reached the social community was the center that do a part of a process highlighted, and the work of the other somehow put aside. This is pretty scary. We won't cope with something like that. I have a bunch of examples, and this is why we got maybe some overexposition through the media for our activity, from our institution. But it's also a clear signal that from the other side, the people are no longer recognize the hospital as the only places where they can obtain this kind of information. And I cannot disclose how the things went, but basically they went to a private company and got what they asked. So once again, let me just thank the people in the lab, the people that do the economical part in my institute, and a really big thank for the organizer and also for the speaker. It was really nice to have a different angle to see the things. Usually many of my work time is in the lab, so I don't look of the whole side of the process, and it's always a really nice experience. Thank you all.